Thank you very much. It's a great pleasure and honor to speak here. Actually, we did not coordinate our talks with Fabian, and maybe we should have, because I'd say there will be some repeats. We use different words. I use the word representation, but you can think of it as latent space. So I will explain again, and maybe this is a sign that it's something important. So I try, I'm part of the people trying to bridge the gap between machine learning, AI, so this field that is moving forward very fast, and there are some techniques, some lessons which have been learned in other fields, in image, text, etc. And certainly this idea of embeddings, representations is a major thing that's happening in machine learning in the last decade, and I'm sure this could be a good way to move forward in a single cell in particular. So I will try, there will be some repeat. I will try to explain my view of what it is and why this could be relevant and how to do that a bit, as well as some challenges. Okay, so the idea, and I will be quick on that, I will not repeat, is that we have an idea of a cell, but maybe we should not talk only of individual cells. Maybe there is a concept of groups of cells, organoids, tissues, etc. So we have some biological objects from which we can collect data, and we want to do many things out of that. We want to understand biology, we want to guess or predict effect of some perturbation, we want to maybe model some, create equations of evolution, etc. And so between the raw data, between the measurements, the experiments, and these applications, we need some mathematical abstraction, some computational framework, and it's possible to, and there is a trend that for each new data, each new experiment, you may decide a particular way to represent your data and to infer something. But maybe there's a way which is a bit more generic, and maybe that's the way to go in the future, which would be to find some representation of your data. And so imagine many possible data representing many different things, and maybe it's possible to embed that in the unified space, which to be decided what it is. Is it vectors? Is it strings? Is it graphs? Is it something else? It's not completely clear, but at least maybe there's a way to represent the biological data into some mathematical framework where then you can perform many tasks. Right, so where does this idea come from? It's a quite natural idea. You've been doing that all the time. We like to visualize data. But in particular, I will insist on what has happened in the last decade in machine learning, in AI, in fields where it works. So certainly computer vision was mentioned already as one of the fields that have been revolutionized by approaches. The ideas are not new. The ideas have been around for several decades, but the fact that it works is a combination of these good old ideas and lots of data and computational power altogether result in quite impressive results. So if you take your smartphone or some self-driving car or many applications, now it's possible to just take an image and perform many tasks. Object recognition, face recognition, segmentation of the image. When you work on videos, you can do extrapolations, predict the future of videos, etc. And so you could imagine that for all these tasks, people have developed specific algorithms, specific ideas, etc. But at the core of it, there is something quite impressive, which is that most of these applications use some representation of the raw input, so the image, as a vector of numbers. Right? So on the top of this slide, you see one of the traditional architecture of deep learning. But if you abstract away what's between the image and the vector, what's important is that people have designed a way to transform some input data into a set of numbers. And from this set of numbers, so each image becomes a vector of numbers. And from this set of images, it's possible to do image segmentation, face recognition, etc., etc. Right? And what's a bit surprising, and what's surprisingly good for companies, labs working on that, is that you basically with the same architecture, once you have more or less found a good way to transform your data into these vectors, this space is super strong at many downstream applications. Right? You don't really need to redesign or reinvent a new representation each time. It's possible to find a more or less strict representation that allows to do many things. So in particular, we talk a lot of latent space, manifolds, etc. I'm not sure in this field people are so excited about the notion of low-dimensional manifold, because the representation here is not really low-dimensional, right? We have observed that if you want to have good products, good softwares that work on images, you need more than 2 or 3 dimensions. You need at least 500 typically, and it will be the same for natural language processing. Right, so here we don't necessarily look at low-dimensional embedding, depending on what you mean by low-dimensional, but the idea that at least there is some representation fixed, which may be big enough to be complex, and when we have more and more data, we need more and more dimensions here, but in this space, then it's possible to work with vectors and perform linear operations. By the way, this image here may look familiar to many of you, but these are not single cells, these are images, right? This is a t-SNE on a data set of images. After transforming each image in the vector space, so as a vector of numbers, you can just apply the same t-SNE or the same algorithms to visualize them. And there is some visual similarity with some of the pictures we have seen this morning. Okay, quickly, there are other fields which have even more dramatically been changed by this idea. Natural language processing, so treating natural language. If you take your smartphone, maybe you've noticed that translation becomes better and better every month. You can do speech recognition. You can almost talk to a computer. When you think of it, you could say, well, we need very complicated model of semantics of the sentence, how to encode the verbs, the construction of the sentence. But the winning technology today is pretty similar to the one used in images. It's a way to transform some input words or sequences into a vector. And then once you have a vector, to do some operation on the vector. So the vector is this set of numbers. And for example, translation more or less amounts to saying, I have a sentence in English, I map it as a vector, and then in this space, where I have also mapped other languages, I'm looking for some sentence in some other languages where the vectors are the same, right? So it seems to be quite naive, because you could say language is complicated, it's hard to believe that you can just encode the meaning of a sentence into a vector, and certainly it doesn't work perfectly, but at least, roughly speaking, this is the winning technology these days. Okay, and so for example, here you can again map a set of sequences into this, so this picture is not as nice as the previous one, it's not a t-SNE, but think that all sentences like half a century ago as coordinates, so here I just show 2 dimensions, but again, in language, typically we use 500 dimensions. We need at least 500 dimensions in order to have a space rich enough to try, to have sufficient diversity to encode the meaning of all possible sentences, and then work on that. Maybe last example, so just to illustrate that beyond, so many applications fields are impacted by this idea. So many people like these days to, for example, represent graphs as vectors. We talk of graphs as one way to represent data, but I would say that the trend, especially in large-scale applications where you have many graphs or very large graphs, is instead to go from graph to vectors, and then once you have vectors, to again do similarity search or do classification or detection of community, etc. So again, it's possible to abstract away the original data and find a space where you can do things. Right, so you could say this seems to be, there is this big trend that seems to pop up in all these fields. What have we learned from these other fields? We have learned that this vector representation, so vector again is just a vector in the sense of mathematics, a set of numbers. So the coordinates, I mean you have data, you map them to some coordinates. Surprisingly, in NLP, in graphs, in images, we have observed that even though it looks a bit naive to represent a complicated object as a vector, sometimes the vector can encode lots of information. So for example, in NLP, we know that we can have semantic operations going from, singular to plural corresponds to some vector operation. So it's surprising, but more or less it seems to be possible to create such spaces. And what we have observed is that this works when you have enough data to train the model and when you design models with sufficient parameters so that the data allow to learn this representation. All right, so what about single cells? So there were already a few papers mentioned. I will just recapitulate some of them. Of course, the field, many people in the field are aware of this machine learning. Fabian is one of them, and there are some other ones. So just to illustrate, this is a list of papers, often published in very nice journals. I think all of them were published in 2018 or 19, so it's a recent trend. But there is the notion, just looking at the titles, you see variational autoencoders, you see deep learning-based things, etc. So people are trying to go in this direction to say, look, we have this data. The data, no, single-cell transcriptomics, for example, are complicated data. You have dropouts, you have batch effects, you have all these things. So maybe the simple, PCA-based dimension reduction are not enough. So let's try to borrow these ideas, the technologies, the models, to learn these latent spaces, these representations. And so most of these papers present very promising results. Fabian presented some of them, so this is, I don't need to go in much details. Maybe I will just mention, for the sake of time skipping this one, but maybe just mention, for example, one paper by a team at Berkeley presenting a software called a CVI. This is just to illustrate that this is a specific paper. There's focus on a specific model, etc. In the paper, you have this picture. It looks complicated if you're a biologist or if you're doing something else. It's hard to understand what it means. But one way to understand that is that in this model, you have a way to take input data. So this would be your single-cell RNA-seq profile. And then the model through some, these are neural networks, etc. So don't look at the details because it's kind of complicated, but you transform the data into a layer here, which is this vector representation. That then you can visualize it in 2D if you like, you can do clustering, you can do many things. But again, there is this idea that we create a specific model that transforms data into representation. This model has parameters, and so we need data in order to fit the parameters so that we learn the representation. And then interestingly, from this representation, you can do many things, like clustering, visualization, batch removal, data imputation, differential expression. So it goes in this direction where you're saying, let's not work on the original data. Let's first transform the data into some space where then, ideally, we have kept the biology, we have kept enough complexity in this space so that we can perform predictive tasks. And maybe this is where we can infer causality. Maybe this is where we can infer the effect of a perturbation, for example. OK, I finished with a few of the many challenges so that people can start thinking about that later. So there's a lot, the idea is, if you want to follow this idea, there are many challenges. One of them is, what are the good architecture when we say, I want a model to transform my input data to some latent space? This is likely to be a bit specific to each data, like we don't use the same models for images, for text, for graphs. So certainly, we may have ideas for models for single-cell RNA-seq, maybe other models for Hi-C, maybe other models for images, etc. So certainly here, there's a lot to be done to take into account the specificities of the input data, of the noise, of the invariants we want to capture, etc. There's a question which is, what is a good objective function? And that's also a question which, when we do PCA or most of the autoencoders, there is a kind of specific objective function, which is we want a latent space that recapitulates the original data. Now, if you look at what people do in language or images, this is not what they do. They try to find other objective functions, other tasks, which sometimes are a bit strange, but which allow to learn robust representations. So I don't have time to explore this more, but certainly overall, I would say that there is a lot to explore in terms of what do we want to optimize when we learn this representation. There are certainly questions about algorithms and data storage, but here I'm not scared that people will find ways to be able to embed billions of cells in a distributed thing. One thing to mention is that if we go in this kind of representation approaches, then it can also help in the notion of how to store the data, how to share the data, etc., because it also provides some format, right? Once you decide to encode your data in one way, then you can potentially store them in that way, and this could be a default format to share data or to combine heterogeneous data. So heterogeneous data is obviously, Fabian mentioned that already, something that comes for free if you decide to go in this unique representation. You could say, I fix my representation, and I want images, RNA-seq, other things to go in these ones, so potentially this could solve these issues. There is this crucial question of what I call biological semantics. So obviously, what I present is very much data-driven. I just say we collect lots of data, and we let the data by themselves find a way to represent a cell as a vector. I have no answer to the question, how do we encode biological constraints? How do we transform things into biological knowledge? But Fabian mentioned that this is clearly a field where many people have ideas on how to move forward. Explainable AI, etc., is typically in these things. And finally, there's an interesting question about what is the level of the things we want to represent. I insisted on cells. Lifetime is about cells, but also cells, a cell by itself maybe is not very interesting. Maybe some lesson learned, for example, in language processing is that a few years ago, people just embedded individual words. So they say, we have a word and we represent a word as a vector. And then when you have a sentence, it's a sequence of vectors. So, we find a way to combine individual words. But nowadays, there is a trend to say, let's directly try to embed a sentence. And in fact, I plotted a 2D picture where you had sentences. So maybe it's possible, and the idea of a sentence is that a sentence is more than a sequence of words. There may be interactions between words. One word can change the meaning of the whole sentence, etc. So the same ideas certainly apply in cells. Maybe we don't, it's nice to work on single cells, but maybe we should think of models to embed sets of cells which may be co-localized, which may be orthogonal. Many things are possible. And so this is also one of the very exciting directions that could be tackled. So I'll stop here. Obviously, there are many open questions, more questions than answers in my talk, but I hope this could provide a view of a possible future for one way to analyze the data. Thank you for your attention.
Thank you, Jean-Philippe. Do we have questions? Please go ahead and use the mic. Thank you.
Very interesting talk. You were showing the power of purely, let's say, data-driven analysis, which is what deep learning does. But I think, as a computational physicist, so not as a biologist, that many achievements have been reached by analysis that include, together with experimental data, the so-called a priori biological knowledge, being in a pathway, being a certain type of cell. So probably one of the challenges, I don't know if there is still an answer, is how to integrate deep learning and purely data-driven models with this biological information, which is at the moment in another part of the latent space.
Yeah, so that's clearly one of the critical and big caveats of this approach. I would say that, one possibility is to hope there will be enough data to learn very powerful things and then a posteriori to relay that to knowledge. This is a bit what we do when we do single-cell RNA-seq, then clustering, and then we realize that this cluster expresses these genes, so we try to make a story, but the clusters are just data-driven. Now, I hope that it's possible also to do a bit more than that. For example, when you design the representation, you may put constraints in the model, and this is likely to be important. Like if I just take the example of images, computer vision works because we use convolutional neural networks. These architectures have been designed because people have thought a lot about what is an image, what are the properties of an image, the fact that you should be translation invariant, that you should be invariant to change its scale, etc. So all this knowledge has been inputted in the structure of the network, and then the free parameters are what are optimized from the data. So I suspect that similar ideas could be put in place for biology, saying that you observe things. Suppose you measure gene level information, you could very well say, well, I have knowledge about pathways, etc., so maybe I should constrain my representation to go through path that would be nodes in this case, but maybe we don't need to do neural networks, but things that would go through pathways which are defined a priori. So that could be one way to do, and at least in other fields, this has been important to include prior knowledge about what is an image, what is a sentence, what is a text, to put some limited constraints on the structure, and then let the data fine-tune the parameters. Maybe that's one way to go.
We have another question here from Fabian, and if you guys are fast, then Giacomo will have a chance.
We try to be short. I like the idea, the last one you proposed about this multi-instance or this multi-object put-together learning. We tried this some time ago for learning just clusters and trying to do some type of better prediction. Didn't work out for us very well. So what type of data would you need? What type of questions could you ask? Do you basically need more cells from one organ? You want to see how they're composed? What exactly would be one of the applications there?
Yeah, I'm not sure, but yeah, I don't have any simple answer, so it can be long, but roughly speaking, I would say there is a question of defining tasks, right? What is your objective? And things like very non-useful tasks for us, but useful for the things could be you hide part of you measure many things on a cell, and then you say, I want to predict half of it from the other half. This is really useless because you don't need that, but this is one way to for example, to include robustness, etc., within cells. Now, in terms of diversity of data, I would say that certainly if we can have, so we need diversity. Having diversity in cells allows to fill the space. So I guess then we can.
And the very last one from Giacomo.
Yeah, so it was very nice. You made a lot of analogies between the real world and the other real world, the transcriptomics, for instance. But you made comparisons, for instance, when it goes about language learning, projecting one language word into a vector and then moving into another language. But so these are similar objects, right? So is that possible? Are people thinking in terms of comparing different types of information? For instance, you would have, I don't know, the transactions, economical transactions, and you could now have on the other side a movie about recording the traffic, okay, how people move, okay? And from that, try to predict analogies and then understand the behavior of a society, okay? And we are confronted with the same thing in cells. We have imaging, and then we have omics. So
this is a very good idea, and this is exactly what people do when they need to do that. So one example would be, for example, companies and labs work on trying to automate the annotation of images. Like you have an image and you like automatically to write a small text to describe the image. And the way it's done nowadays in Google and other places, globally, is to say, well, we have images, we can embed them, we have text which can be embedded, and finding a text that explains an image corresponds in the feature space to find a good matching between a text and the image. So this is just one example where you use one representation for two data that look very different, but you find a way to align the representations. So that the semantic, if you think of it as a semantic space, they express the same thing visually or in terms of text. Yeah.